Welcome to Academic Jeopardy!
Hi, my name is Bernice Krieger from Monroe High School, and this is Genetics Jeopardy! Let's play! The categories are Mendelian genetics, Punnett squares, or meiosis. Okay, and the choices are A, law of dominance, law of segregation, and law of DNA. B, law of dominance, law of DNA, and law of independent assortment. C, law of dominance, law of segregation, and law of independent assortment. And D, law of genes, law of DNA, and law of pea plants. The answer is C, law of dominance, law of segregation, and law of independent assortment.
Mendelssohn came up with three important conclusions. That the inheritance of each trait is determined by factors that are passed on to descendants. 2. That an individual inherits one such unit from each parent for each trait. And 3. That a trait may not show up in an individual, but still can be passed on to the next generation.
Let's continue playing Mendelian genetics. Mendel took two true breeding pea plants with contrasting phenotypes and bred them. The F1, or first filial generation of phenotype, showed A, DNA of P1, B, the dominant trait, C, the recessive trait, or D, mutations. The answer is B, the dominant trait.
A dominant trait is one which will be expressed if one of the parents has the gene for that trait. A recessive trait is one that will be expressed only if both parents carry that trait.
Okay, Mendelian genetics, again, for 300 points. The law of segregation states that each parent in the P1 generation can give A, only one allele to the offspring, B, only recessive alleles to the offspring, C, both alleles to the offspring, or D, mutated alleles to the offspring. You're right. The answer is A, only one allele to the offspring. Each parent can only donate one allele, one from the egg and one from the sperm, to make a diploid. Next question, Mendelian genetics for 400 points. And the question is, Mendel took two pea plants, one with a recessive phenotype for flowers, being white, the other with a dominant phenotype for purple flowers, and the genotype is unknown in the purple flowers because it could have been a hybrid, having one purple trait, one white trait, or having pure two purple traits. So he bred them, and the F1 generation outcome was all dominant phenotypes. So what was the genotype? A, capital P, small p, crossed with capital P, small p, giving us two capital P's. B, two uppercase P's, crossed with an uppercase and lowercase p, giving us two uppercase P's. C, an uppercase p and a lowercase p, crossed with two lowercase p's, giving us two lowercase p's. Or D, two uppercase p's, crossed with two lowercase p's, giving us a hybrid, or an uppercase p and a lowercase p. Well, you're right. It's D. Two capital P's, that means pure purple, bred with two lowercase p's, that means having two alleles for white, and giving us a hybrid or heterozygous capital P, small p. Next question is Mendelian genetics for 500 points. A brown-furred squirrel is mated with a gray-furred squirrel. All offspring, or F1 generation, shows patches of both gray and brown fur. 2 F1 squirrels are bred, and the F2 phenotype ratio is 1 brown squirrel, 2 patchy squirrels, and 1 gray squirrel. This is a case of A, complete dominance, B, incomplete dominance, C, codominance, or D, mutation. The answer is C, codominance, which gives us 3 different phenotypes. Another example in humans would be wavy hair, where a straight-haired person has offspring with a curly-haired person, and you get a wavy-haired person. So that's an analogy.
Codominance refers to a relationship between 2 alleles of a gene. It occurs when the contributions of both alleles are clearly visible and do not overpower each other in the phenotype.
Punnett squares for 100 points. A Punnett square uses the possible alleles from the P1 or parent gametes to determine A, the phenotype probability only of the F1 generation, B, the genotype and phenotype probability of the F1 generation, C, the DNA of the F1 generation, or D, genotypic probability only of F1. Ah, you're right, B, genotype and phenotype probability of F1.
The Punnett square is a diagram that is used to predict an outcome of a particular cross or breeding experiment.
Assume that the tall trait in a pea plant is dominant and the short trait in a pea plant is recessive. A capital T small t or hybrid plant mates with another hybrid capital T small t. What will the genotypic and phenotypic ratio of F1 be? Okay, we know this as a hybrid cross. You're right, the answer is A 1 to 2 to 1 ratio, where it's 3 out of 4 cases look tall and 1 case looks short, but the 1 to 2 to 1 ratio means that 1 out of 4 of the offspring will be purely tall, 2 out of the 4 will be hybrid tall, and 1 out of the 4 will be purely short. Punnett squares for 300 points. The question is, assume the superpower of laser beams is a recessive small b. A laser beam hero, 2 lowercase b's, mate with another superhero of unknown genotype or phenotype. The F1 offspring phenotype is 2 no beams to 2 beams. What was the genotype and phenotype of the unknown parent? The answer is D. Capital B, lowercase b, and that would equal no beams at all.
Genotype is an organism's full heredity information, even if not expressed. Phenotype is an organism's actual observed properties, such as morphology, development, or behavior.
Punnett squares were 400 points. The question is, how many different allele combinations would be found in the gametes produced by a pea plant whose genotype was capital P, small, lowercase p, and 2 uppercase r's? Okay, that means that we're crossing 2 traits, and so how many different allele combinations could there be? The answer is A. We have 2 possible combinations, the uppercase P and the uppercase R, or the lowercase p and the lowercase r, and the only reason for that is there's no lowercase r to be found in this genetic combination. So you can only use an uppercase R, but you have an uppercase P and a lowercase p. For 500 points, Punnett squares, the question is, A Punnett square showing the result of a dihybrid cross, meaning two traits, dihybrid, okay, meaning they're both hybrids. So capital R, lowercase r, capital Y, lowercase y mates with capital R, lowercase r, capital Y, lowercase y, and shows that the trait for P shape, round or wrinkled, and P color, yellow or green, and in the case of a P, it's, well, we'll see. Okay, it's yellow is dominant. Okay, so the choices are A, are always homozygous, B, are linked, C, are independently assorted, and D, have the same traits. It's the law of independent assortment. Mendel said that everything was inherited separately. Okay, we found out later that certain traits are linked, like, for example, people that have arthritis tend to also have psoriasis. The outcome of meiosis is what? Okay, and the choices are A, two haploid gamete cells with different DNA alleles, B, four haploid gamete cells with differing DNA alleles, C, four diploid gamete cells with differing DNA alleles, or D, two diploid gamete cells with identical DNA alleles. The answer is B, four haploid gametes with different DNA alleles. Okay, now we're up to meiosis for 200 points. The two phases of meiosis are A, meiosis 1 and mitosis, B, meiosis 1 and cytokinesis, C, meiosis 1 and meiosis 2, and D, prophase and metaphase. And who knows the answer? You're right. The answer is C, meiosis 1 and meiosis 2.
The outcome of meiosis is a gamete. The cells are haploid in number.
Okay, we're going to do meiosis for 300 points. And the question is, the outcome of meiosis 1 is A, the duplication of homologous chromosomes into tetrads, which divide into two genetically identical? Wait a second. Yep, it says identical diploid cells. B, duplication of homologous chromosomes into tetrads, which divide into two genetically differing diploid cells. C, the formation of four haploid gamete cells, or D, the formation of four identical diploid. And who knows the answer? Okay, the answer is B, duplication of homologous chromosomes into tetrads, meaning four, which divide into two genetically differing diploid cells. And the next question is meiosis for 400 points. And the question is, the difference between mitosis and meiosis is A, in meiosis 1, two non-identical genetic cells are formed, and in meiosis 2, four haploid cells are produced. B, in meiosis 1, four identical genetic cells are produced, and in meiosis 2, two identical haploid cells are produced. C, in meiosis, there's no interphase and no cytokinesis, and D, in meiosis, there is no chromosome duplication. And this is a tough question because the answer is, who knows the answer? It is in meiosis 1, two non-identical genetic cells are formed, and in meiosis 2, four haploid cells are produced. Okay, we're up to the last meiosis for 500 points. Okay, you have to fill in the blanks for this one. Meiosis starts with blank cell and finally ends with blank cells. This process is used in order to produce blank. All right, so it starts out with, well, you're going to be thinking diploid or haploid, ends up with diploid or haploid, used to produce, what is it used to produce? Is it used to produce a regular cell or a gamete? So let's see what we have here. Okay, it starts out with, okay, A, 4 haploid, 2 diploid, and new cells. B, 2 diploid, which ends in 4 haploid, used to produce gametes. Okay, that's a possibility. C, 1 diploid, 2 haploid, and body cells. Is that what meiosis is about? Body cells or sex cells? Okay, and finally, D, 1 diploid into 4 haploid with gametes. Okay, and who knows the answer? Okay, the answer is D. I'm going to read the fill-in for you. Meiosis starts with 1 diploid cell and finally ends with 4 haploid cells. This process is used in order to produce gametes. And that's the end of our session today for academic Jeopardy for genetics in preparation for your CST exam.
Thank you for playing Academic Jeopardy with Bernice Krieger.